Hello, is this better? Can you guys see me? I hope this one is better. If this one didn't work, we go sleep. I don't know what happened. This is the first time I'm experiencing that stuff. I hope this one is better. It's not better. Sleep straight. I don't know what happened. Like everybody, so I was busy talking all this while, and nobody heard me. Hey. So let's allow everybody to come in so that we can proceed. So sorry about this. So sorry. So what was the last thing you guys heard me say? Let me know where to start because I don't want to start again, please. Excitability. Oh Lord, that's like beginning now. I'm even thinking that excitability, neuromuscular junction. Hey, I'm afraid of sharing that slide again. But all this while it has been working very well. About the motor unit. I think I should risk it again. I hope. I hope it doesn't happen again, please. I'm praying it doesn't happen again. So we are discussing muscles, all right? We've already talked about. Okay, yeah, okay, good. Okay, that works. The old video is there for anybody to watch the introduction. So calcium, this is very straightforward. I don't want to waste time on this. The more question you answer, the better. Calcium that is pushed out by the action potential that is passing through the T tubule rushes inwards, holds the door, that is the troponin, holds the handle of the door, that's troponin. Then the troponin moves the door, that's tropomyosin. It's a complex troponin-tropomyosin complex. Then it's when it opens the door, that's the active site of actin. Then myosin can now lap to actin. So this purple one is myosin, trying to lap, waiting for this guy to open the door so that it can lap to the active site. Please, I hope this thing does. If it lags, please, if you just notice that it lags before it stops completely, tell me, let me unshare, and we just jump into the questions. This means that it doesn't want us to discuss it completely. Then you can just go watch the video. All right. So that's what happens. All right. That's what contraction is about. For relaxation, it's just the opposite. All right. Everything starts detaching. Like calcium has to leave. When immediately it gets the ATP. You understand? It leaves the handle, and the handle shuts the door, and immediately shuts the door. The relaxation starts occurring. So I don't want to spend so much time on it. So this is relaxation. Other things you need to know is the length tension relationship. You need to know what rigor mortis is. You already know what rigor mortis is, but you need to study it again to learn more about rigor mortis. You need to know what the myogram is. Threshold, twitch, you have to know what they are. So I really want to run off these things so that, yes, you need to know what recruitment is. You have to know what recruitment is. Then you need to know what TREP is. That's 10 to 20 stimuli. You understand? 10 to 20 stimuli, that's TREP. This is tetanus. Remember the difference between tetanus and strep is the number of the stimuli and also the TREP. They return back completely to the resting point. But incomplete tetanus does not return to the resting point. It's keep increasing. Complete tetanus is, this where it stretches and fatigue sets in. All right. Incomplete tetanus is 20 to 40. Mind you, 20 to 40. All right. So that's incomplete tetanus. Why complete tetanus is 40 to 50. You need to know the difference between isometric contraction, isotonic concentric, and isotonic eccentric. All right. Isometric, isotonic. Once it is not moving, it is isometric. Once it is moving, it is isotonic. Now, if it's reducing in size, right, muscle shortens with tension, it is what? Isotonic concentric. If muscle lengthens, it is isotonic eccentric. I hope this thing is not. Is it lagging? Can you see my hand? Okay, I think it's not lagging. Please let it not lag. Okay. So let's see. We can see you. Okay, thank you. Okay. Yeah, you need to also know the phosphagen system. That's phosphagen system. This thing is phosphagen system, all right? When short intense exercise, 100-meter dash, oxygen need. Yeah, hello. Welcome. Oh, God. So oxygen need is briefly supplied by myoglobin for a limited amount of aerobic respiration at onset, rapidly depleted. So this muscle meets most of the ATP demand. So the question is, how does it create ATP? There are two enzyme systems. We have the myokinase and we have the creatine kinase. For the myokinase, this is ADP. This is another ADP. What myokinase does is it will take this ADP. All right, it's diphosphate. That's two phosphates. It will take one phosphate from this guy and put it on this guy. So this guy now becomes what? ATP. Why this guy becomes AMP, monophosphate? This one becomes ATP. I don't know if you understand. That is action of myokinase. Sorry, let me drink the ATP first, please, so that we have energy to talk to you guys. Water is life. Oh boy. So that's the function of myokinase. That's what myokinase does. Why creatine kinase? It does the same thing, but in different way. It obtains its phosphate from something called phosphate storage molecule, known as what? Creatine phosphate, CP. All right. So that's what we call, the two of them is what we call phosphagen system. All right. When you need a burst of energy to do something, that's phosphagen system. So this is myokinase. You see ADP changes to AMP. Why it takes that P and adds to this guy here, it becomes ATP. This creatine kinase, we break down this creatine phosphate to creatine, then take the phosphate and add to this ADP and make it what? ATP. So that's how they work. It's as simple as that. All right. So let me see. Oxygen depth replenishing. Yes, you need to know about the slow oxidative, slow twitch, red type 1 fibers. It's the same thing. They can ask you slow oxidative, SO. They can ask you slow twitch. They can ask you red. They can ask you type 1 fibers. All of them is the same thing. And the beauty of this one is that, it has a lot of mitochondria and myoglobin. Remember, why do you tire out? You tire out because when you run, your muscles start working on anaerobic respiration. And anaerobic respiration produces what? Lactic acid, which tires your muscle out. All right. So, but in this particular type of muscle, all right, they have a lot of myoglobin. And myoglobin contains what? Oxygen. So it does not tire out easily. So a lot of people that run long race, you understand, long distance, they have slow oxidative switch, red type 1 fibers. All right, because they have a lot of oxygen in those muscles. Now we have the one we call fast twitch or white or type 2 fibers. All right. So for people that are so fast, all these Usain Bolts running 100, 200 meters race, that means they have very strong combination of fast twitch muscles or type 2 fibers or white fibers or fast twitch fibers. All right? That's what you need to know there. You will see a question on it, probably. Okay. So this is summation. This is resistance training. This is endurance training. Now, other type of muscle. All this while we've been discussing skeletal muscle. This is cardiac muscle. Cardiac muscle is limited to the heart, where it functions to pump blood. Properties of cardiac muscle. This is what you need to know. All right. Most important thing you need to know about cardiac muscle is that it's striated and it is what? Involuntary. Striated. All right. And what is causing this striation? Intercalated disc. And it has a lot of what? Electrical gap junctions, which allows each myocyte to directly stimulate its neighbor, unlike the skeletal muscle that does not. All right. So cardiac and smooth, they have these gap junctions. Okay. Voluntary, it has a pacemaker, that's the SA node that can make it, have auto-rhythmicity. That's ability for it to what? Beat on its own. All right. And because it beats on its own, it has, it keeps beating. Your cardiac muscle can never get tired. If it gets tired, the person dies. It's as simple as that. All right. So it's rich in myoglobin and what? Glycogen. And it has a large abundance of what? Mitochondria. You understand? Now, smooth muscle is another very good one, all right, that you need to know. The calcium needed for muscle contraction comes from what? ECF. Skeletal muscle, where does the calcium come from? The calcium comes from the sarcoplasmic reticulum. Smooth muscle, where does it come from? ECF. Cardiac muscle, how about cardiac muscle? Cardiac muscle has pacemaker. Smooth muscle gets their calcium from ECF. I am just repeating it. All right. And it's capable of mitosis and hyperplasia. I hope you know the meaning of my hyperplasia. I'm not saying it. No striation. Smooth muscle, no striation. All right. But they have fusiform shapes, but no striation. And they are involuntary as well. And they also have what? Gap junctions. So smooth muscle, cardiac muscle, they have gap junctions, but skeletal muscle does not. So if somebody has a pathology that affects gap junction, that means the smooth muscle and the cardiac, we have problem, but the person's skeletal muscle will not have a single problem. That's the equation. So these other things you need to know. Let me see if there is something. Peristalsis. Okay, this is dystrophy. Types of dystrophy. This is myasthenia gravis. Now you need to know why despite the fact that you are not doing pathology, but you need to understand the concept of myasthenia gravis. All right, it's an autoimmune system disease that attack the antibodies attack what? Neuromuscular junction. All right, and bind ACH receptors together in clusters. So they disarm the ACH receptors, and when they disarm the ACH receptors, what happens? The ACH will not be able to bind. And once the ACH is not able to bind to the receptors, that means the channels will not be opened. And if channel is not opened, like sodium and potassium will not enter and go out. And if they don't enter and go out, action potential will not be formed. And if action potential is not formed, nothing will push off the calcium. And if nothing pushes off the calcium, that means it will not bind to the troponin. If it doesn't bind to the troponin, that means the tropomyosin will still remain in place, covering the active site of actin. If the active site of actin is covered, that means the myosin will not even be able to lap. If the myosin is not able to lap, there will be no what? Contraction. You need to understand this in this sequence. And if there is no contraction, that means there will be paralysis. And which type of paralysis? It will be what? Flaccid paralysis. I told you there are two types of paralysis. We have spastic and we have flaccid. All right? Spastic, very tight. Usually happening when somebody has a upper motor neuron lesion. Usually, if it's caused by lesion. But this time around, it's not caused by lesion. Instead, it's caused by what? ACE receptors not being functional, just like type 2 diabetes, because that's exactly what happens. Insulin is being produced, but the insulin receptors are not functioning. And what do I tell you about is the receptors? The receptors are the bouncers and in the door. Insulin, ACH, these are hormones and neurotransmitters, and they serve as what? First messenger. Chicken. And how do you treat it? Cholinesterase what? Inhibitors. Why do we want to inhibit acetylcholinesterase? Because usually acetylcholinesterase is what breaks down acetylcholine into acetyl and choline during relaxation, and they go back. All right. So this time around, we don't want to break it down. We want to leave a lot of acetylcholine, all right, in the neuromuscular junction, so that If there is a peradventure, any receptor that is still free, it will be able to see enough what acetylcholine to bind on it. So it's just a common sense. All right. So that's myasthenia gravis. These things it causes droopiness, weakness of muscle. That's what I call flaccid paralysis. All right. You couldn't be able to even gaze upwards. Okay, that is it. I hope you guys understand this thing properly. I hope you understand, most especially you understand this connection that we discussed here about entering and going out. Please, I want to quickly remove this thing before it hangs again. Why am I having PTSD? Why am I having PTSD? Is it only 4 that I'm seeing? Please, people should raise this thing to like 20. If you've not click it. All right. I don't know why I'm laughing this night though. I'm laughing too much. Somebody will say Nchebe is so happy. As if there's a time when Nchebe is not happy. Okay. Are you guys ready for the questions? Yes. I need to get like 20 years. Keep saying yes. Keep writing yes. Please, the rules, once again. When we ask question, everybody should attempt. Even if you think, oh, that you don't know it, pick one of the answers. Then if I didn't give option, please write not sure. It's extremely important that you do that, please. Like I really need you to do that because it encourages me, to continue talking, to continue as vibrant as possible. All right. Don't just keep quiet and look. And another thing is that when you attempt it, it's easy for you to remember it. When you're in the exam hall, you'll be like, oh, I attempted something like this. I am sure this is what I picked. But he said, no, that's not the answer. This is the right answer. Or I attempted it and I got it. And if you have something you need me to explain, please put 911. Hold on, just give me a moment. Muscular tissue. Multiple choice. Okay, let's test the microphone. Testing, 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 testing. Testing, testing. Options. A, B. Just hold on, let me put the options, all right? Oh, what's happening now? B. I don't know why everything is so slow this night. C. D. We are just testing microphone now. I hope this microphone does not spoil very early. Okay, how many have I posted? A, B, C, D, then E. So the question, drugs called calcium channel blockers may be used to lower blood pressure by causing arteries to vasodilate. How do you suppose these drugs work? A, they prevent calcium from entering the sarcoplasmic reticulum of smooth muscle. They stimulate the calcium pump in the smooth muscle, thus removing calcium from the calmodulin. They prevent calcium from exiting the sarcoplasmic reticulum in skeletal muscle. They prevent calcium from entering cardiac muscle, thus slowing down the heart rate. D, they prevent calcium from entering smooth muscle, thus allowing the muscle to relax. Come on, guys. What's your answer? Come on. Everybody. Come on, come on, come on. Activity, activity, activity. Let's push it. Let's push it. You have to be able to pick an answer, please. Yes, you guys are very correct. I'll try to be very fast today, all right? It's okay. You guys are very correct. The answer is they prevent calcium from entering smooth muscle, thus allowing the muscle to relax. Remember, they are asking you about causing arteries. This is arteries. Arteries are smooth muscle, all right? Arteries are smooth muscle. So it's that straightforward. So this is just testing the microphone, guys. Testing the microphone. Testing. This question. That's the question. A, is this? B, is incoming. B, C, is this? And D, A, B, C, then D, is this? E, is this? Okay, so which of the following is an accurate comparison of the skeletal muscle to smooth muscle? A, smooth muscle. Why is this thing not too bright? Perfect. Oh, let there be light. Okay, which of the following is an accurate comparison of skeletal muscle to smooth muscle? Smooth muscle contracts more slowly but relaxes more rapidly. Smooth muscle contracts more rapidly but relaxes more slowly. Smooth muscle contracts and relaxes more slowly. Smooth muscle consumes more ATP to maintain a given level of muscle tension. Smooth muscle depends on an axon to supply the calcium needed for contraction. Come on, guys. Let's go. Who is not talking? We have 47 people here. Who is not talking? Come on, please. Everybody. Everybody. Somebody said the life is frozen. Is it still frozen? No. No, it's no longer frozen now. I think it's better now. So you guys are very correct. Smooth muscle contracts and relaxes more slowly. I don't think I need to explain these questions. If you think I need to explain it, please just tell me. Okay, it's not frozen. Thank you. Just tell me and I'll explain. Why is the concentration strength of smooth muscle relatively independent of its stress? No, this question doesn't make sense. Which muscle can contract without the need of further stimulation? Okay, look at this question, guys. There are three muscles that we discussed about, three types of muscles we discussed about. So I'm not giving you options. Those three are your options.